Hi, everyone. My name's Rob Shields, and I've been seeing a lot of different pieces of information, a lot of misinformation currently during the pandemic regarding the mRNA vaccines. And I just thought that it would be helpful for me to use my unique skills because I'm not a doctor, but I have been talking to friends and family members who are doctors. I do have a background in biology and I'm an AP biology teacher. So I thought maybe I could bridge that gap between what some of the doctors are saying about mRNA vaccines and us as the general public. So I wanted to give everyone a little bit of a clearer understanding of how vaccines work in general and how the new mRNA vaccine is intended to help us fight the coronavirus. So let's get started. The way that a typical vaccine works is a little bit different from the way that an mRNA vaccine works. So, a typical vaccine will actually contain a dead or weakened form of what's called a pathogen. And a pathogen is just a fancy term that we use for any illness causing substance. So, you would think of a virus or a bacteria as this pathogen. And the presence of a pathogen causes the cells of the immune system to react to it and to produce antibodies. Now, this is a somewhat of an oversimplification of the immune system because there are many moving parts to the immune system, but essentially, there are important factors that the presence of a pathogen will trigger. And all of those have to do with the surface of the pathogen, and on its surface it has these proteins. And so the antibodies and other parts of immune cells will bind with those proteins on the surface of the pathogens, and they'll actually signal to other immune system cells to destroy it. And so when people become vaccinated against a disease, they won't typically get sick from it because their body now has a quicker ability to recognize it because they are able to produce antibodies much more quickly as a result of new immune system cells that have been produced. We call them memory cells. So there are these specific memory B and memory T cells that recognize the pathogen a lot quicker than next time. And those memory B and memory T cells are part of a greater group of cells that we consider, when normal people are talking about it, we call them white blood cells. So there are special types of white blood cells that the vaccine has given the ability to remember a pathogen. And then when the pathogen returns to the body will be ready for it. The immune system will recognize it more quickly, and so you won't get sick from it. That is how a typical vaccine works, meaning it has a weakened form of the pathogen. But mRNA vaccines are a little bit different. And in order for us to discuss how an mRNA vaccine works, we kind of have to discuss the differences between RNA and DNA. And RNA and DNA are actually very similar, but there are a couple of differences. So one of the main differences is the sugar, and the sugar on RNA is ribose, whereas the sugar on DNA is deoxyribose. And so that really doesn't make much of a difference when we think about it, but what you'll understand is that RNA stands for ribonucleic acid, whereas DNA is deoxyribonucleic acid. So it does have a different sugar, and it does have a different nucleotide base. So if you look at the series of A's and T's and G's and C's that code for all of the proteins in your body, RNA substitutes a uracil for a thymine. But again, not a big deal in what we're talking about here with the vaccine. What is important is this last bullet, which discusses how RNA is single-stranded and DNA is double-stranded. And you can see that. So the RNA has the single strand here, whereas the DNA has two strands. And RNA will typically be a copy made off of DNA, and it'll be a small copy because DNA contains many, 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 many different genes or series of bases that code for a protein. And RNA just has a snippet of it, and that little snippet we call a gene. Each little gene is a code for one specific protein. So your body makes a lot of different RNA molecules because you need to make those proteins. So DNA has the code, but the RNA is going to send that message. So DNA is found in the nucleus, and it contains all of your genetic information for proteins, but RNA is similar to DNA because it also carries genetic information. However, DNA is much larger, and since it's so much larger, it can't leave the nucleus. So the DNA will stay in the nucleus, but the message for creating the proteins has to get somewhere. So the mRNA, which stands for messenger RNA, which is much smaller and single-stranded, will actually be the messenger. It will leave the nucleus, whereas the DNA won't, and it'll go to the ribosomes so that proteins can be made. So if you look here, these abbreviations are your amino acids, and they're getting put together to make a protein. You'll notice that here in the ribosome, that's this brown thing, it has the mRNA, which is represented by this red band moving through, and it has another type of RNA, which is tRNA, called transfer RNA, that matches up with the codons here and helps create this protein. And so really what we need to understand here is that the messenger RNA is what's used to create the protein, and it brings the information to the ribosome, which tells the ribosome the order to build the protein out of its building blocks, which we call amino acids. And when all of the amino acids are assembled in the correct order, you make a specific protein. If the amino acids are put in a different order, you make a different protein. So the order is very important, and the order is coded for here in the RNA, which the RNA is usually getting that message from the DNA. Once the RNA has delivered this message, there are enzymes in the cell that will destroy the RNA, and it'll be gone, and it'll be reused. All of these nucleotide bases will be reused and the formation of new RNA. So when we talk about the coronavirus, a lot of times you'll hear it called SARS-CoV-2. And it's called SARS-CoV-2 because it's so similar to the original SARS virus, but it's not the original. It's a second, slightly different version, so it's SARS-CoV-2. You've probably also heard of the disease called COVID-19. That stands for coronavirus disease 19. And why the 19? It was discovered in 2019. So that's where COVID-19 comes from. But when you hear coronavirus, you're actually hearing a conversation about a group of different viruses. And all of those different viruses, when you look at them under a scanning electron microscope, produce a halo. And so if you look here on the left, I have this picture of a halo, which actually reminds me a lot of the sun. And so that's actually where the name came from, because this halo is very similar to the sun's corona. And so that's where you get the term coronavirus. It's a virus that has this halo, this corona. But the halo is really just the spike proteins that you're seeing. So when you've seen this more common image of the coronavirus or SARS-CoV-2, you can see all these spike proteins, and that's what's showing up under the microscope as the halo, and that's why it's called the coronavirus or SARS-CoV-2. It's got this halo and these spike proteins. And the spike proteins are really important for the virus's ability to function. So what the virus will do is it will actually bind with receptors found on the cell membrane of the cells. And it's basically like a lock and key. And unfortunately, because the spike proteins match up so perfectly with the receptor, they're able to enter the cell. And once they're in the cell, they can use their messenger RNA located inside of the virus to replicate itself, and it can make many, many copies of itself. Obviously, this is problematic because it can be damaging to the cells. And now that it's replicating, there are more and more coronaviruses that will infect a person. So in order to stop this, we have these two different messenger RNA vaccines. And these are all of the ingredients that those vaccines have. There are two different types, right? There's the one from Pfizer BioNTech, and there's the Moderna vaccine. And they're very, very similar. There are slight differences in the salts that are found there. And the salts are used to provide a better body chemistry so that the vaccine kind of matches up better with your own body's pH, your own body's concentration of certain salts. And they're also there to help stabilize the vaccine. It wouldn't make sense if the vaccine broke down prior to being able to do its job. So these are the ingredients, and they're pretty straightforward. There are a bunch of different types of lipids, but they have similar functions that we'll talk about here on our next slide. So the purposes of all of these ingredients is that, first of all, you need the mRNA, because this mRNA is the code for the spike protein. People get nervous when they hear, oh, you're going to inject a vaccine filled with messenger RNA from the virus itself. But don't be nervous, because it's not all of its messenger RNA. It's just the specific code for how to make a spike protein. So you can't get sick from just a spike protein. There's no viral information there for anything else. So it wouldn't be able to replicate itself or do anything. In this case, it's just the code for the spike protein. But the problem is messenger RNA, because it has ribose and ribose has a charge, can't enter the cell. It'll actually get blocked by the cell membrane because the cell membrane has an attraction to it. And so it can't diffuse into the cell. So in order to allow the messenger RNA to enter the cell, we have these lipids. And the lipids are these fats, basically, that protect the messenger RNA so that it doesn't break down before entering the cells, and also so that it can enter. And so that's what the lipids will do. When people are getting these allergic responses to the vaccine, they're typically having that reaction to the lipid. Something about the lipid is causing their body to recognize it as a foreign invader, and they're having this adverse reaction. However, if you're there and you can get epinephrine, it should help you. You're not going to gain any type of like lasting illness from that. And then, so like I said, you have these other salts and substances to help stabilize it or stabilize the pH. And lastly, there's sucrose. Think of like table sugar, right, that you might put into your coffee, something like that. So it's just sugar here. And the use of the sugar is that actually prevents the lipids from getting stuck to each other. So preventing them from sticking to each other is really useful, obviously, because the vaccine wouldn't be able to work if it was all clumped together. So that's what the purpose of the sugar is. But most importantly, it's the messenger RNA and the lipids that not only protect it, but allow that messenger RNA to enter the cell. And so, like I said, the mRNA is only the code for the spike protein, nothing else. And so when the messenger RNA enters the cell, it goes to those ribosomes I was talking about. That's what RNA does. It goes to the ribosomes. So a lot of people are nervous because they think the messenger RNA might enter their nucleus and change their DNA and turn them into some type of crazy mutant or something crazy like that. But that's not how it works. The messenger RNA will never go into the nucleus there. The messenger RNA from the vaccine goes directly to the ribosomes. And the ribosomes, which are responsible for making proteins, in this case, are going to read the instructions and put the amino acids together in order just to make the spikes. So now all of these coronavirus spikes we've been talking about get created, and the body starts making more and more of the spikes, but again, only the spikes. So there's no virus here. It's just making more of the spike protein. Then the cell sends those spikes out into the bloodstream, and those cells from the immune system recognize those spikes as foreign invaders. And when the invaders are recognized, they create antibodies, they create memory cells. And so these memory cells, if they ever see this spike in the future, will make more antibodies to react more quickly to the coronavirus. And why is that important? Well, the most common way that these immune system cells are going to see the coronavirus in the future is the coronavirus is going to be there with all its spikes. And so it's going to be able to attach to those spikes, and it's going to be able to signal an immediate immune response, which should give the person who receives the vaccine immunity from the virus. And so this would be what it would look like. You'll notice these multiple binding sites for the antibody. It's able to bind to the coronavirus and signal other immune cells to come and destroy it. I've got a simplified version of just any virus that has the specific area where the antibody is binding, but that's the gist. So your body is not replicating the virus. It's not changing your DNA. It's just using the messenger RNA that we've created from the genetic code of the coronavirus to make spikes. And once it makes all of the spikes, the body's able to recognize it as a foreign invader, create memory cells, and create antibodies to prevent you from getting sick again. Thank you so much. I hope that was helpful. As I've always said, always please consult with your medical provider. Talk to your doctor about your situation before moving forward. Don't just take my word for it. Make sure that you're consulting with medical professionals. But hopefully this gives you a better understanding of how the mRNA vaccines work and how you can prevent yourself from getting sick with a deadly disease.